Today we're going to start talking about how chromosomes are related to genetics. So Mendel didn't know about chromosomes or meiosis. Everything that he came up with was based on the phenotypes of the offspring in his pea plants. So when he came up with his principles and he said there was a certain characters that masked other characters, and he also said that one parent would give one copy even though they had two copies of each character, he didn't know that those characters were called genes and they were on chromosomes and that they were separating during meiosis. So today we have what's called the chromosome theory of inheritance, and it says that genes are codes on your DNA that are found at certain loci or locations, positions on your chromosomes, and that Mendel's principles or laws of independent assortment and segregation, those laws are happening to your chromosomes during meiosis. So Mendel's laws are related, are basically about the chromosomes. So Mendel's laws, these were his three laws. So the way that the chromosomes orient themselves, the orientation in metaphase 1, they want to know what principle of Mendel's does that have to do with? Well, that has to do specifically with independent assortment. That the chromosomes could line up if we have like big B, little big C, little c, and those are your chromosomes. They could line up like this, or they could line up like this with big B, big C, little c, and that it was completely random. So the way that those chromosomes lined up side by side, you could have the big B on the left and the little b on the right, or vice versa. And the fact that they separate at anaphase, meaning, remember, you have all these pairs, but you're randomly going to give just one copy from each pair. So big B, you're going to give either big B or little b, not both. This is Mendel's principle of segregation. So the chromosomes segregate during meiosis at anaphase, and the fact that they can be oriented with either chromosome on either side, that's independent assortment. And this is showing the same thing. Again, Mendel's laws and how when the chromosomes lined up, it could be the big R and the big Y, or it could be the little r with the big Y, or the big R with the little y, that there were all these variations in how the chromosomes could line up. That was independent assortment. All right, so it turns out that there were some studies done when organisms were crossed, and they predicted that 9 to 3 to 3 to 1. That's the one you would get with the dihybrid cross. So like if you had big B, big R, big B, big R, little r. If you recall, on our previous lecture, we talked about how 9/16 would have both dominant traits. 3/16 would be dominant for the first trait, but recessive for the second trait, etc. Well, they found genes that didn't show this. That when they did the cross, they didn't come out with that outcome, and they wanted to figure out what was going on. Why didn't that work? And it turned out it was because the genes were what were called linked. The genes were on the same chromosome. In other words, normally we would expect, in this case, if you did this particular cross, that you would get a 9 to 3 to 3 to 1 ratio. So this is basically a prediction. So 9/16 of the total, 215, you would have expected 71, 71, and then 24 with recessive for both traits. That's what you would have expected if the traits were on different chromosomes. But that's not what happened. What ended up happening is you had a lot more of dominant for both traits and a lot of recessive for both traits, and not a lot with these in-between, what are called recombinant phenotypes, like different combinations than what the parents had. Anyway, we're not going to get into linkage too much in here. All you really have to know is that when genes are linked, they're stuck together on the same chromosome. So look, in this case, big P and big L sort of stick together, and little p and little l stick together, which means when meiosis happens, you don't get sperm that are big P with little l or little p with big L. Those two possibilities don't end up happening, and so the outcome comes out different than what you would expect. So, for purposes of our class, all you really need to know, linked genes are genes that are on the same chromosome. They do not follow Mendel's rules of independent assortment because, remember, if the chromosomes separate independently of each other, well, sure, then if P and L are on two different chromosomes, you could get any combination of big P with little l, big P with big L. But if P and L are on the same chromosome, then when the chromosomes separate, you're always going to get big P with big L together and little p with little l together. And remember, you have thousands of genes, but you only have 23 different chromosomes, right? 46, but 23 pairs. So you know some genes have to be linked together on the same chromosome. So don't worry too much about the results of this experiment. Now, the thing is that if the genes are linked, you might wonder, well, then how would you ever get a different combination? Like if red hair and freckles are linked, then everybody should either have red hair and freckles or brown hair and no freckles. How do you get somebody that has not red hair, but they do have freckles if those genes are on the same chromosome? And the answer would be crossing over. Because if, by some random chance, you have red hair and freckles on this chromosome, and let's say brown hair and no freckles on this chromosome, but during meiosis, these two swap places, guess what? Now red hair and no freckles are on the same chromosome. So crossing over would allow new combinations that you would not have seen because of the genes linked. Now, there's a way to measure this. It's called recombination frequency, and we're actually not going to spend a lot of time talking about that. Just know, again, linked genes on the same chromosome, you intend to inherit them together, but when crossing over happens, you could get what's called a recombinant phenotype, where you do get a new combination that didn't exist before. This is showing, again, how you can actually calculate that, and they can actually map how far apart the genes are based on how much they recombine. This was in a fruit fly experiment. And then they can actually figure out the distance between genes on a chromosome and create what's called a genetic map or a linkage map that would show where the genes are located on chromosomes. But again, you're not going to be tested on that. You do need to know about sex-linked traits, though. So if genes can be linked to specific chromosomes, it turns out that genes can also be carried on specifically the sex chromosomes. So let's review what a sex chromosome is. So in mammals, furry things, right, like us, we have XY for males and XX for females. Now the Y chromosome is not the same as the X. The Y chromosome actually causes you to develop male, but aside from that, it doesn't have a lot of other genes on it, to be honest. And so for any trait that's not on the sex chromosomes, if it's on the autosomes, which is basically any of the other 44 chromosomes you have, aside from the 2 sex chromosomes, those are non-sex chromosomes, and everybody has 2 copies of those. But what if a gene is actually carried on the X, the sex chromosome X? Well, that would mean that a female, since she's XX, would have 2 copies of that gene, if the gene is B, let's just say. But a male who only has one X, he would not have 2 copies. He would only have a single copy of that gene. So this is going to make a big difference when we go to do a cross. If we cross males and females and a gene is on the sex chromosomes. Now, other species have different ways of determining sex. Birds have something called a ZW system. And in some reptiles, the temperature actually determines whether the babies hatch male or female. So above certain temperatures, maybe they're all male. Below a certain temperature, they're all female. I think that alligators and sea turtles have this type of sexual development. So it's not, don't assume every living thing has males and females the same that we do, that it's defined by the chromosomes. But animals, vertebrate animals and mammals in particular, do have the XY system. So King Henry VIII was always blaming his wives because he kept having daughters. Turns out it wouldn't have been his wives' fault because his wives would have been XX, right? So when they go to make eggs, they can either give X or X. That's it. But King Henry, being a male, he would have been the one that could have given X or Y. So the mom every single time would have given an X, and if he kept having daughters, that would have been his fault. He would have been the one giving that second X. In order to have a son, he would have needed to give the Y chromosome. So this is what they look like in actual pictures. Notice how teeny tiny that Y chromosome is. I think it only has about 24 genes on it. You do not need a Y chromosome, obviously, or females would not exist. You do have to have an X chromosome, though. You cannot live without an X. So there's no such thing as a guy who has like 2 Ys. You would not be alive. You have to have an X. It's got a whole bunch of things on it that are really important. And this is showing, again, a male makes sperm. They can either have an X or a Y, and then there are the 22 other chromosomes. So sperm has a total of 23. And then the female, the mom, she has also 46 chromosomes, 44 that are autosomes. They don't determine sex. And then the sex chromosomes. She's going to give 23 in the egg, 22 autosomes plus only an X. That's the only option. And then if the sperm that fertilizes that egg is a Y, you get a male. If the sperm is an X, you get a female. So sex-linked traits are specifically carried on the X chromosome. Theoretically, they could be on the Y, but there's almost nothing on the Y. So usually we call them X-linked genes because they're typically on the X chromosome. 2 really common ones that code for diseases in us would be color blindness. The ability to see color, red and green in particular, is on the X chromosome, and the certain blood clotting factors, that if you're missing them, you have a disease called hemophilia. Those genes are also found on the X chromosome. In fruit flies, this is the way actually sex chromosomes were first discovered. It turns out that the white versus red color in fruit fly eyes was determined by a chromosome, a sex-linked trait on the X chromosome. Again, the Y chromosome has almost no information. Typically, it makes you a male. That's pretty much what it does. Now, if a sex-linked trait was on the Y, who could inherit it? Well, pretty much only guys could inherit it. And if a guy had it, so if you had XY and then some kind of a trait on the Y, he would basically give it to all his sons. Every son he had would get that Y chromosome from him that carried that bad trait. But he would never, ever give it to a daughter, right? Because he would give his X to make a daughter. So this is showing, again, a little summary. So sex-linked traits, they're genes typically on the X. Most are recessive. There are some sex-linked traits that are dominant, so I added the word most there. Fewer females tend to be affected, and that's because females have 2 X's. So in order for a female to show the trait, she would actually have to get 2 bad copies of the gene. But since males only have 1 X, they can't be like heterozygous. They can't be a carrier. So what they're showing here is this is a normal female. This is a female that's a carrier. They're using X with an X little X minus. What I would oftentimes do is use a letter instead. So like use XN for normal vision. Let's say XN, X little n. So now she's carrying the gene for colorblindness. But she's not colorblind because that's recessive, and she's got a normal one, so we call her a carrier. A female that's colorblind would be X little n, X little n. Now, male can't be a carrier. He only has 1 X. So if he's X big N Y, he's normal, and if he's X little n Y, he would be colorblind. So that's sort of what these look like. So you're always going to draw the trait, the letter for the trait, and you're going to actually put it on the X. So in fruit flies, X big R codes for red eyes. See how they write the gene on the X, and X little r codes for white eyes. And they literally can have red colored eyes and white colored eyes. And here's an example of a cross. Now with these crosses, it looks a little different. So they're showing the male, he either gives X little r or Y. He's a white-eyed male fly. Now notice how they only made 1 X here. You could do it with 2. So she's X big R. I had taught you guys to cut your Punnett squares down so you didn't have to make 4 for a cross like this, but you could. So it's done the same way, and then you put your X's back together. So this is a female, normal vision, female, normal red eyes, sorry, male, red eyes, male, red eyes. So everybody would have red eyes in this particular cross. But look at this one. Now we have a female, she's a carrier, and the male again has red eyes. Check this out. So when we do this cross this time, we see that our females all have red eyes. I don't like the way they did this. I always write the capital letter first, so big R, little r. But we have a 1 in 4 chance of a male with white eyes. So in the next lecture, I'll do a few more of these practice problems with setting up these sex-linked crosses, but that at least gives you an introduction.